in the room to have you join us for today's Friday seminar series session. Obviously, we've got a bunch of competing demands today between the regular things having to do with this point of the semester to a beautiful day outside to something called the NCAA tournament. So obviously people could be pulled in a bunch of different directions, but I'm thrilled that so many of you have set aside time today to not only highlight the work of our colleagues in athletic training, but also to share with our larger community some of the work that our faculty and students are doing in the areas of traumatic brain injury, otherwise known as concussions. We've had the great fortune to be part of the university's group that's received a large bolus of funding in this area from the Department of Defense over the last 3 to 5 years. And then we've actually been able to build on that with some additional grants and other grants that are currently under review. But I think for those of you who know a little bit less about this work, you're going to find that it's not only very interesting, but quite multidisciplinary. And I think will give all of us kind of a moment of pause, you know, the next time we're watching a basketball game and somebody bangs their head and what's going on in the moment and what the subsequent sequelae of that can be. So we're fortunate today to be joined by our colleagues, Dr. Jamie Mansell, Dr. Ann Russ, who's joining us from Omaha, Nebraska, Dr. Jane McDevitt, Dr. Geoff Wright, and Dr. Joon Park. And they're all going to give relatively brief presentations on their work. If you do have any questions, please put them in the Q&A feature. I will then come back at the end and go ahead and kind of moderate that Q&A and discussion. And I'm really hoping that not only does it give all of you an opportunity to learn more about this really interesting work, but also think about potential collaborations with this group of investigators going forward, because I think this is an area where we can continue to highlight our excellence and be part of the fabric of the growth of the research mission of the college. So with that, I'm going to turn things over to Dr. Mansell and Dr. Russ, who are going to kick us off by talking about the management of concussions by athletic trainers.
All right, thank you so much. You can see the full slide screen, right? All right, so thank you so much for joining us today. We really appreciate, again, on the Friday afternoon. First, I want to make sure I see a lot of athletic trainers in the audience, so I wanted to give a quick shout out to everybody. Happy National Athletic Training Month. Our theme this year is providing healthcare everywhere. Perfect time with March Madness and perfect time to deliver this concussion talk as well. So Dr. Russ and I are going to talk today a little bit about the clinician side of concussions. And we're going to kind of try and level the playing field here so that everybody understands some of this more basic information about definitions and how we evaluate concussions, and then also how we can rehabilitate and return folks to activity or sport. So we're kind of really setting the stage for the research that will be presented by the following speakers. So our learning objectives here, we're just going to identify the current concussion diagnosis and evaluation techniques, describe those current trends in management, and then we'll also discuss some of the state law as it relates to concussion. So to make sure we're all on the same page, we have just a few definitions here. Concussions are a traumatic brain injury induced by biomechanical forces. They could be a result of a direct blow to the head or an impulsive force. And these forces cause a rapid onset of typically short-lived impairment in neurological function. And they're considered a functional disturbance, not a structural deformity. A few important things to note. These may or may not include loss of consciousness, and they will not show up on imaging. And we've found those are two of the most common misconceptions that we see in some of our patients and some of the parents of our patients. So we really wanted to make sure that we got that message out here today. For the most recent epidemiological data, we're seeing that there's been a steady increase in concussion rates across the United States. And while we're unsure as to the reason why, it's speculated that it's because we have better education for patients, athletes, parents, et cetera, and therefore there's better recognition. So folks are coming forward with their signs and symptoms, or they're coming forward after they have these forces and being evaluated by their clinicians. Knowledge is really rapidly changing. And unfortunately, we may have some clinicians that are not as up to date as we would like with contemporary information. And they might not be using the correct information for diagnosis and rehab and return to activity. Unfortunately, this can result in misdiagnosis and mismanagement. Some of these old school, quote unquote, things that we're seeing are grading of concussions. This was done decades ago and was based primarily on loss of consciousness. And again, from the last slide, you know that we don't need that loss of consciousness for it to be considered a concussion. We see that some clinicians might be using scans to diagnose, so CT scans or other imaging. And again, a concussion is not going to show up on that imaging. And we also see some folks saying we should wake the patient every hour, but just like the rest of our bodies, our brains need rest to repair. We also see people returning athletes or workers back to activity too early, and really generally not seeking out or incorporating those best practices. We, as the athletic training faculty, do a presentation on a specific type of vestibulo-ocular function to assess concussion. And people love this talk. It's active. We have them up, they're practicing the skills. And at the end, when we ask how many are going to incorporate that into their clinical toolbox, we definitely do not see all the hands in the room raised. It's a free, quick assessment. By the end of our talk, they all know how to do it, but there's still some sort of a barrier where they're not incorporating what they then know are the best practices. Because the signs and symptoms are so varied, a multimodal technique is needed for a full evaluation. We have a list here on the screen about some of the different types of testing that should be incorporated, and we need to make sure that we're hitting all of these different aspects. And we need all of these evaluations because the signs and symptoms are going to vary so much between patients. And I'm going to turn it over to Dr. Russ to continue on.
Well, thank you for all that, Dr. Mansell. As we saw, there's a lot of different things we need to do with the evaluation piece to see what's happening in these different buckets of concussion. I had a alum from our program describe it that way once, and it really jumped out to me and Dr. Mansell. So there's some very numbers, but we went with the 6 main categories of presentation. There's cognitive fatigue, vestibular issues, ocular issues, post-traumatic migraine, cervical presentation, and also anxiety and mood considerations. So even though there's these 6 categories, people might experience more than one bucket of concussion signs and symptoms. And when we look at the evidence, we see migraine and headaches as one of the most common signs and symptoms. Anxiety and mood disturbances, 28% of people report that. 20% of people report vestibular issues, 14% ocular issues, and then 12% cognitive and fatigue issues. So when it comes to the recovery piece, and Dr. Mansell, could you advance the slide, please? Thank you. What we see with this is that recovery can vary. So when we look at the normal time frame, about 80 to 90% of adults are going to recover within 2 weeks, maybe even some as soon as 7 days. And in children, that time period is longer. It can take up to 4 weeks. And when we look at a breakdown between ages, kids between the ages of 5 and 12 recovered faster than adolescents who were between the ages of 13 and 18. In addition, with that same population, we have to consider recovery in their school aspect as well as their physical activity. So looking at high school students, about half of them will miss up to 11 days from their sport after concussion. We also see some time that might be missed in the classroom. And about 30% of athletes will take longer than 14 days to unrestricted play. So even though we can have that 2 to 4 week guideline, there are people who are going to go beyond that, and that's okay. We do see some differences in recovery between males and females. Females on average take about 3 days longer than males to recover. And new evidence points to using early aerobic training and other management strategies to decrease the days that people are symptomatic and to decrease the days it takes to return to play. So we really want to promote that active recovery that corresponds with the person's concussion presentation. So here we see a guide to managing different concussion presentations, and each concussion presentation has a different subjective and objective measure, so what the person's experiencing and what we can measure. And they also have different responses to a graded aerobic exercise test, which is proposed as a necessary assessment in order to determine the best course of treatment. So what's recommended is that we use the Buffalo concussion treadmill or the Buffalo concussion bike test. And this is safe to do 1 to 9 days after they've sustained the concussion. Different types of concussion are going to respond differently to that aerobic training. And identifying how that patient responds, we can put them into different management options to help them recover from their unique concussion. It is important to note that the Buffalo concussion treadmill and bike tests are both safe. They don't increase concussion signs or symptoms the day after testing, so it's good for us to add to our toolkit. And what's, again, really important is that we have to target the intervention to the patient's presentation. If, in the past, it was a one-size-fits-all approach to concussion management, we know now that's not going to benefit our patients. We have to target what the patient is experiencing difficulty with. And that does include working with interprofessionals teams that have specialties in those different areas. So our last learning objective was about state law. And in 2009, Washington State was the first state to establish return to play concussion laws. Other states soon followed, and now all 50 states have legislation. Specifics about return to play can vary from state to state, and that can include the population covered. For example, there's some states that only cover athletes in a public school. There's other states that cover all minors engaged in physical activity. It varies in who can diagnose concussions, as well as what the clearance is for return to activity. What's interesting is that 8 states have also identified the need to support students post-concussion. So we have 8 states with return to learn legislation, and that legislation outlines what supports and resources a student may need in the classroom and offers guidance to schools on what that should look like. What's important for all of us to keep in mind is that these laws only establish the minimum of care. So it's like, at least this is what we should be doing. So people might choose to go beyond that. But the other thing that is challenging is that these laws that are establishing the minimum of care might not be up to date with emerging concussion evidence. We've learned a lot more about concussions since 2009, and a lot of the research that we've seen is from after 2009. So the other thing to keep in mind besides state law is that if you're working with an athlete, keep in mind that different sporting bodies may have concussion regulation that's maybe beyond what state law is. And so that's something that you might have to be aware of. And then you'd want to make sure that you're following that regarding clearance and return to play protocols. What it all comes down to, what's our clinical bottom line for our clinicians, is that we have made massive gains in concussion knowledge over the past decade. And some big takeaways is that concussions can present in many different ways, even subsequent concussions in the same individual. Because of that, we have to use a holistic approach for concussion recognition, diagnosis, management, and recovery. We have to center that care around the patient's needs, and what's best for the patient is to use an interdisciplinary team that's appropriate for their needs. So athletic trainers can be involved in education, evaluation, recognition, and return day-to-day management and return to play. Physical therapists can be used for targeted interventions like vestibular therapy. In a school setting, there's nurses, social workers, school counselors, and educators that all have to work together. Public health professionals can evaluate risk factors and look at solutions to minimize risk. Physicians, physician's assistants, and other providers, as recognized by states, can provide evaluation and diagnosis. Occupational therapists can assist with re-engagement with day-to-day activities, and speech language pathologists can incorporate can work with cognitive communication deficits such as attention, memory, language, and executive functioning skills. So thank you for hearing our segment on clinicians' approaches.
So Dr. Mansell and Dr. Russ, thank you both very much for your time and a great kickoff to this session. I am going to reserve questions for the end, but as we transition to Dr. Wright's talk, I'm going to actually just give you a comment to think about. I'm thrilled that you put up the slide at the end about state laws. And, you know, as you know, I'm a big fan of this work, and I'm thrilled that you guys are having the opportunity to go regionally and nationally to share not only this, but some of the other great work that you guys are doing in the area of sexual harassment with athletic trainers. But perhaps when you're back, Dr. Russ, we should introduce you guys to some of our colleagues in HSAP, Health Services Administration and Policy, because they do an awful lot of work at looking at issues like state laws and how they impact healthcare. And given your local expertise in this area, you know, maybe there's some opportunities for some of our graduate students to take those two disciplines and bring them together and do some work in this space, that because it is, you know, it sounds like this is really a moving target at the state level. But for those of us who know somebody who suffered from a concussion or you know, follow athletics, we know how pressing this issue can be. So when you're back and have some time, you know, feel free to reach out to me, and I'll connect you to some people in HSAP. So, but otherwise, thanks and enjoy Omaha.
Thank you.
All right, so with that, I'm going to turn things over to our colleague, Dr. Geoff Wright from the Department of Health and Rehabilitation Sciences and the Division of Physical Therapy, who's going to share with us some of the great work that he's been doing, looking at concussions, primarily in military populations. Dr. Wright?
Yeah, thanks, David. Let me just see if I can screen. All right. So I do actually do a lot of work with athletes, but since I know Jane McDevitt is going to be talking about a good bit of that, and I'm surrounded by a lot of athletic trainers, I figure I'll focus more on veteran and military relations. I've been working with. I hope my connection is stable enough that you can hear me. I've been working with the VA and in the military for a number of years, and I've also been working with virtual reality. And virtual reality is, as you all know, is a to bring in, but it's also a way to measure kinematic, measure the movement of the body through the head move. Somebody's wearing something like a head-mounted display. So we've been developing technology in my lab that uses portable virtual reality to test people's balance and also test their visual and ocular function. And right now we're focusing on traumatic brain injury and post-traumatic stress disorder. So I'll talk a little bit about how these two might relate. This is funded by the DMRP, the Congressional Directed Medical Research Program, and also supported by the local VA. But it also has been supported by the Syracuse VA, where I did a research sabbatical with them. This current project really is starting to test more of the ocular motor and vestibular function, whereas we've been out of balance testing for quite some time. And so taking a look here, we can see wearing a head display, standing on a phone surface. This can affect someone's balance, and viewing this twirling scene back here. Really all we need to make this portable is to put all custom design software in a head-mounted display. So we've done a whole series of validation tests to make sure that head tracking is comparable to other measures of balance and kinematics, and we've gotten very high Pearson correlations. We published this in Human Factors. So I'll be mentioning a number of publications we've had over the last decade, with some of the more recent ones that are supporting why we're pursuing this particular topic of looking at TBI. So mild traumatic brain injury, remember, is often used interchange the word concussion. And though concussion is sort of a subset of mild traumatic brain injury, I'll probably use them interchangeably. Now, what's interesting about TBI and mild traumatic brain injury in the military is that it often co-occurs with post-traumatic stress disorder. Because if you think about a traumatic event that may have resulted in someone getting hit by the blast wave from a bomb thrown against a tank or thrown to the ground, usually there's other traumatic things going on in a setting like that. So we see these occurrences of TBI, and we see post-traumatic stress disorder going along with it. And we often don't know what symptoms are being caused by what, because they have a series of overlapping symptoms that make it very hard to tease apart. Now, for the clinicians in the group here, the importance of actually accurately diagnosing what the underlying etiology is extremely important, because it's going to dictate how you then treat it. So if you have a psychogenic problem, so something that's related to psychology or psychiatric disorders, or physiogenic, something that's related to physical problems that may have occurred due to these two things need to be separated. And if you can separate them effectively, then you can start to treat them. And this has been a problem for clinicians for years, is knowing whether something is PTSD or TBI related. I mentioned a bomb blast. There's actually a series of ways that one can be injured by being in the vicinity of bomb blast. It doesn't always involve being hit directly by actual also we call that blast TBI, and that's one of the primary causes of traumatic brain injury in military. I won't go too much into the details of the coup and counter coup. That's your standard, your sports concussion. Let me just go back one slide. The tertiary, if you on the slide here, the primary is the blast wave. The secondary can be injury can be due to penetration or impact from bomb fragments. And then the third is being propelled by the blast wave and then thrown against a solid object or the ground. That is the injury that's most comparable to a sports injury. So we do a lot of research on athletes as an injury mechanism for a group of individuals who wouldn't have necessarily PTSD that goes along with their TBI. And so we can use them as a control case for a military individual in the military who potentially has a TBI that could be associated with a trauma that could cause PTSD. I won't go too much into, I know that the time is limited, so I don't go too much into the details of the military, but it's very similar to sports concussion. The numbers see for military actions in early 2000s, and most of these are classified as mild traumatic brain injury, so over 80%. In the US population, compare that to sports concussion and all types of concussions, we're about 2 to 4 million annually. And the military obviously is a much, much smaller population, but among them, there's a large percentage are getting exposure to mild traumatic brain injury. And so in the veterans, we see PTSD also occurring in as many as 400,000 since the military actions in the early 2000s. And there's not any good clear biomarkers for diagnosing PTSD. And this is similar to MTBI, concussion, is that it's all symptomatology based. But the symptoms that tend to overlap with TBI and PTSD include memory loss and concentration issues, depression, irritability, anxiety, and dizziness and loss of balance can be associated with both of them. So there's some evidence that suggests psychological factors rather than physiological factors can also account for many of the post-concussive symptoms. there's a timeline that one can look at. So the recovery from an injury from a TBI has a series of timelines. Sometimes it's with minutes to hours or within days. And then you have many other things that include the inflammation, post-inflammation, axonal injury, apoptosis, so cell death, demyelination and remyelination and neurodegeneration that can happen over years. So there's these various timelines. So there are symptoms and signs that can arise many months or years after the injury. So again, we are really in need of good biomarkers and good tests for determining where the injury signs and symptoms are at after the injury. So why am I focused on balance? These are good biomarkers for neurological injury, and this is known for over 100 years because of how difficult it is for a person to stand on 2 feet. Balance is known to be a marker for some sort of underlying injury. Some of the comments that I alluded to in this talk, hopefully my internet again is stable, is that we see these sequelae that are like dizziness and vestibular and visual problems. And so focus in on those. And an aspect that is of importance is the 3 senses that are involved here are not just vision, but also not a sensory need for balance can be affected by traumatic brain injury. Because if you look over these areas that under the main function are involved in balance and postural control and dizziness, there's numerous areas throughout the brain and the cortex, cortical areas, brainstem that are involved in here. So if we had a counter coup of the head after a concussion, the full damage, the level of damage could injure many areas of the brain. So again, focusing on balance, we've designed some custom design balance tests that include looking at a virtual scene. So this is the TV-based version that we did a number of studies on. We're funded by a DoD project about 10 years ago. That lasted a few years, and we got a lot of data on both athletes and military and veterans. And we were able, through a series of tests, making the tests more and more specific and targeted, we were able to determine that if someone is placed in a situation where they have a visual then deficits then are healthy controls. I am going to speed through these results because I've given them in many talks, but some of y'all probably seen them already. We used to receive operating characteristic curves to test sensitivity and specificity, very significant levels of detection between healthy versus concussed. We have done binomial regression to kind symptoms like balance and vestibular ocular work that I did with Jay McDevitt and Ryan Tierney and others with my team. And we saw very, very high levels of areas under the curve. So very high levels of sensitivity and specificity. We tested this on but actually the history, just having that cut in the last 10 years, more ones, 2, 3 more, this showed a big difference, just concussion, multiple, where we could tease them apart in this most challenging balance condition of visual and vestibular conflict. So last couple slides, we're now looking at PTSD and TBI. We did a study that we just published recently. It's actually press right now in the Journal of Neurotrauma, where we looked at, again, the history of concussion in these complex multisensory manipulations, where we had over 50 veterans that we tested, and we saw that, again, the number of concussions was a significant factor in that visual vestibular conflict condition. So that brings us to this final project that I mentioned at the beginning of my talk, where we're looking at ocular motor tests, including vergence insufficiency, dynamic visual acuity, so visual and vestibular tests that specifically look at the vestibular system. And we're designing these in a head-mounted display that with custom design software that allows us to make a portable test that could be used anywhere in any sort of environment, and a nice objective test. So the level of experience of your clinician becomes less of a factor. So inter-rater reliability goes up, intra-rater reliability goes up when you've got a really good, reliable measure that can take away this unreliability that may occur from your 20-year experienced ATPT clinician versus your fresh out of school ATPT clinician. Obviously, there's a lot of variability that can go into subjective pen and paper tests, but if you can make a nice subjective, quantifiable technological solution, then it might help with assessment of your athletes or your veterans or your military folks. So I'll finish with that. A lot of, I hope I gave full credit to all the people that have been involved, and this is just a partial list, and we've had a lot of funding over the years, including ARL, as David mentioned, CDMRP, and veterans, and obviously Temple has been extremely supportive. So I might have gone over by a couple minutes, I apologize, but I think I just...
No, you did great there, Dr. Wright. Unfortunately, you broke up a little bit in places in your slides, and your voice got a little bit wonky, but I think we got the gist of things. And as we pivot over to Dr. McDevitt, one comment that I'll make for people, if you do ever get the chance, and I suspect that all of our speakers today would be more than happy to give you this opportunity, to go and see how they do some of this testing and some of this work down in the basement of Pearson McGonigal, I would really encourage you to do it. It is really, really fascinating stuff. Something that's, so very different than so much of the work that we do in the college. And, to see how they make these assessments and so forth, I think for all of us who are researchers, it's something that we have an opportunity to learn from. But I also think, also for collaboration, because I think as we've already seen in these first two talks, there is that overlay between the physical symptoms that may be observed in somebody who's suffered a traumatic brain injury and the psychosocial issues that either are being worsened because of the TBI or are part and parcel of the recovery process. And I'm sure we'll come back and talk a little bit about that during the Q&A session. So with that, I am going to have us pivot over to Dr. McDevitt, who, as you could tell from Dr. Wright's talk, has collaborated with Dr. Wright and others, and she's going to talk with us a little bit more about some of the genetic associations that she's seeing in her work.
Great, thank you. So concussion recovery is quite variable, as we've been seeing through the previous talks. The NCAA CARE Consortium found that in the collegiate population, it can be around 16 days, where Tamura et al. Found that in a high school population, it's a little bit longer. Younger athletes need a little bit longer time, so it's about 20, but plus or minus 24 days. Though the average or quote unquote normal timeline has been fairly well defined, prolonged or persistent long concussion has not been well defined. There are many concussion recovery factors, such as concussion history, the frequency of the head impact itself extrinsically, but then there's also intrinsic risk factors, like we said, age, sex. One risk factor that has not been definitively parsed out is genetic variation. Genetic variation is likely to determine concussion recovery because genes code for those proteins that are going to be involved with neuronal recovery. If that code changes, it could change the structure of that functional protein, and it could alter how that protein can actually help and promote healthy neuronal recovery. So if we just step back and look at how a concussion occurs, obviously we have this head impact that causes this acceleration, deceleration. And if you take a look at this little gelatin here, you can see that there's going to be a little bit of neuronal stretch or neuronal strain. The amount of this neuronal strain is important. If it is stretched above 10 to 20% beyond its resting length, that's where we're going to see this pathophysiological process, or this this functional process. So again, it's not a structural, we're not seeing any actual tearing of the neuron, but what we are seeing is a cell environment complication. And this was termed the neurometabolic cascade by Giza and Hovda. And if we take a look at the important proteins that are involved with neuronal recovery, we can take a step back and look at what genes are then going to be important to code for these proteins. So let's just dive into this pathophysiological process just briefly. So first, we have this depolarization from the neuronal stretch. That's going to allow excitatory neurotransmitters such as glutamate to be released in the synapse. One of the most common receptors is the N-methyl-D aspartic acid channel that has the NR2A and NR2B subunit. Those are the glutamate subunits. This channel, when activated, allows calcium into the cell. And similar to any time calcium floods into the cell, that will allow the depolarization to be prolonged, as well as further depolarizations to occur. And when we aren't getting this signal for normal nervous signaling, the brain will go into a sense of, okay, we're out of homeostasis. So who do we turn to when we need to return to this homeostasis? We're going to go to the sodium-potassium pump. However, what do we need to activate the sodium-potassium pump? ATP. What's the problem after a brain injury? There's no ATP. All of the local blood flow, all of those surrounding capillaries were also torn or sheared during that mechanism. So there's no blood flow, no ATP. So what's plan B? Well, powerhouse of the cell. Thank you, mitochondria. Mitochondria are going to help and eat up this excess calcium. Problem there. Have any of you ever experienced overeating? What happens? You become bloated and you're a couch potato. Same thing here. The poor mitochondria takes up too much calcium and it becomes bloated and dysfunctional. So we still have this, these depolarizations, this hyperexcitotoxicity. At the same time, the axon itself is also stretched, and that causes further allowance of calcium to come into the axon. And it also allows the axon to become compacted. So now we have some axonal swelling, and now we can't get more mitochondria or more proteins to the cell body to help with recovery. So if we take a look, we have plenty of proteins that are really necessary for neuronal recovery. Now, if we work backwards, we're going to evaluate what proteins are necessary and choose those genes. So we're going to take a look at the NMDA channel, and we're going to take a look at the NEFL neurofilament. Some other things that I'm not going to go into detail on, but I do want to just put out there in terms of how do we do genetic association research, is that the location of this variation on the gene and the variation type are really meaningful. So just briefly, where this variation occurs within the gene is necessary to note. So many geneticists evaluate the promoter region, because this tells us and this allows how much of that protein is going to be produced, right? How much of that is going to be transcribed and then translated into that functional protein. The other bit that geneticists focus in on is the exon region. And the focus is primarily on the exon region because the exon region carries the code to which will develop the genetic code to which will develop that mature protein. We do not focus as much on the intron, though there is a lot of research going in the world of epigenetics in terms of what the intron's responsibilities are. But in terms of evaluating how can this worked into recovery and concussion recovery, typically, as you can see, the introns get spliced out. So there's nothing in the in the mature protein with the intron. So then if we look at the variation type, there's many different types of single nucleotide polymorphisms or repeat polymorphisms, which are basically different types of genetic variations. Again, they can occur in that non-coding region, the intron, or in that coding region, the exon. And then it can be similar or not similar to that coding amino acid. So again, choosing the different type of variation is really important. In my talk today, I'm going to be talking about the promoter variations and looking at not a single nucleotide polymorphism, but repetitive repeat polymorphisms. So typically, how do I do this? So like I said, I'm going to choose my my gene here, and this is just a little snippet of how you can kind of think about where in the grand scheme are you coding this from. So this is a map of my SNP here. So, and this is the locus, which is basically just the location of the SNP within the whole entire genome. So 16 means that this is on the 16th chromosome. P refers to the petite arm in comparison to the long arm. Band 13, subband 1, subband 3. So if we go to that location and blow it up, you can see just prior to exon 1, about 479 nucleotides upstream is my GT repeat polymorphism. So in the sport concussion lab, which again, anybody is able to see us in action, we will go through a post-concussion protocol, and then we will follow those athletes prospectively until full return to play. Again, the prolonged recovery is not well defined, but in our population, we decided to draw the line in the sand at 60 days. So if they took more than 60 days, they were prolonged. If they returned within normal, within 60 days, they're then termed normal. And then we collected the saliva, and from the buccal cells that were then within that saliva sample, we blasted open those leukocytes and pulled the DNA from the nuclei of the leukocytes of that white blood cell. And then we were able to genotype the repeat promoter polymorphism with capillary electrophoresis, where we basically just said, do they carry the common or popular polymorphism, or do they carry the disease-causing or rare polymorphism? So just wanted to, again, briefly review. We are, one of our works was in response to evaluating the neurofilament light. Again, the neurofilaments, microfilaments are really important in the scaffolding of the neuron to allow communication and axon transport. If that code allows that neurofilament to become flimsy or too stiff, that could increase the risk of a concussion. Similarly, Seidler et al. Found that after repetitive head impacts on animals, they found a decrease in axonal transport due to that neurofilament compaction. Therefore, proteins that would be necessary for neuronal recovery to enter into the cell body, like mitochondria, were not able to do so. So again, that's another reason why NEFL was a good protein to find a genetic marker regarding. And we published this work in the Journal of Neurotrauma. Again, there are many different types of proteins, but this is just this one work. So we found that carriers of the short allele GT repeat polymorphism were 1.5 times more likely to recover in 60 or more days compared to those carrying the long allele genotype. And this is similar to what they found with carriers of disease-causing NEFL rare alleles. They found more or an over transcription of NEFL, which contributed to the stiffness, which then increased the risk of these neurodegenerative diseases such as Alzheimer's, Huntington, Charcot-Marie tooth disease. Another one of our focuses was on that NMDA channel, and there are two glutamate receptors. Remember, glutamate is that excitatory neurotransmitter that consistently depolarizes the neuron, leaving the neuron in a state of vulnerability if they were to get another hit or another stretch. So that NR2A is one of the glutamate subunits produced via the GRIN2A gene. And again, when activated, these cells allow calcium to come into the cell, and that allows it to lead to excitotoxicity, or too much calcium, allowing for too many depolarizations and never allowing that neuron to return back to homeostasis, which could alter recovery. So we published this work in brain injury, and what we found here was that the long repeat, 25 or more GT repeats, was associated with being twice as likely to recover in 60 or more days compared to those carrying the short GT repeat. Again, in comparison to previous works, who also found the long GT repeat to be associated with poor outcomes, such as more likely of suffering with Parkinson's, schizophrenia, bipolar, or having smaller brain regions by carrying the long repeat polymorphism. So there are limitations to genetic studies in these specific studies that I, just briefly introduced to you today. The low N, it's less than 100 student athletes. We focused on very few genes and few SNPs. There are a lot more targets to evaluate. However, we did identify that there are target genes within the neurometabolic cascade that we can evaluate to further refine our concussion management strategies to identify where are these problems with recovery occurring. Hopefully, we'll be able to continue with larger prospective or case control studies, possibly a genome-wide association study. There's only been one genome-wide association study in concussion to date, and it's heavily limited based on their ability to state the control. So it was looking at concussion risk, not differences in concussion recovery. And why is this work important? This work is important because we need to individualize our concussion management. There are a lot of problems with identifying best practices in safe return to play. Therefore, understanding their genetic risk could be helpful in the concussion management, as well as educating them on the risk that they are taking under by participating in something like football instead of tennis. So they can we can provide further education and counseling to anybody that has an increased risk for concussion or prolonged concussion recovery due to their genetics. I could not do this without so many people, so I just wanted to give a brief and big shout out to everybody here. And we'll take questions at the end. Thank you.
Well, thanks, Dr. McDevitt. And along those same lines, for anybody who does have questions, feel free to start putting them into the Q&A feature, and I'll moderate the question and answer session after Dr. Park's talk. Dr. McDevitt, though, I really enjoyed listening to you for two reasons. One, it brought me back to college biology, hearing your enthusiasm talking about neurotransmitters and the gaps and all of that stuff. And it reminded me how much I loved learning about that stuff originally. And the second thing, I love the fact that you had an overeating analogy in there. So when I think about my obesity work, I'm like, oh, this resonates with me. So with that, we're going to pivot over to Dr. Joon Park, who is also part of the Department of Health and Rehabilitation Sciences. But many of you probably don't see him a lot because his physical lab is up on the Health Sciences campus. But I know he regularly comes down for departmental meetings and to meet with students and to be in the classroom. And he's going to tell us about the important work that he does. And I apologize, Joon, I just got you wrong. You're in kinesiology, not HRS. So he's in our Department of Kinesiology. So we have multiple departments and divisions represented here. But with that, Dr. Park, go ahead and tell us a little bit about your work.
Okay, thanks, David. Actually, it is really truly interdisciplinary research, and there are lots of things to be done in near future, hopefully. So today, I'm going to focusing on emerging biomarker for concussion. And I will specifically focus on the really, really small, tiny particles that you can find in the blood, which is called extracellular vesicle or nanoparticles, as a potential biomarker for sports-related concussion. So what is extracellular vesicle? Extracellular vesicle consists of large particles and small particles, but both are still submicron, so it's a nano scale. So larger particle is called a microparticle, smaller particle is called exosome. But altogether we call it, extracellular vesicle or EVs. So extracellular vesicle were first found in about 70 years ago, right? 60 years ago. And first they just term it as a cell dust because literally all of different type of cells spit it out. And actually these EVs are so tiny. So back then there's no technology exists to really study on what is the function of EVs. Actually, there are plenty of EVs circulating in our blood. Actually, 10 to 8 to 10 to 13 EVs per ml of plasma sample exists. So there are so much things that we can analyze and play around with it. The size is again just submicron. It is ranged from as small as 30 nanometer to about 1 micron. So that is actually the size of 1 millionth of a size of a hair. So it's really, really small. And actually this extracellular vesicle contains a lot of different molecules. It has nucleic acids, a DNA or RNA, or also proteins. So basically our research is analyzing those cargo molecules after isolating EVs from blood plasma. So how we analyze it? Actually, there is a machine called flow cytometry. Actually, Becton Dickinson BD company first coined this about 50 years ago, but it's very, prototype, very old type kind of machine. Actually, the resolution, the scale is way above 1 micron, so 1,000 nanometer. So with the traditional flow cytometry, there's no way really you can measure the number or characteristics of EVs. But still, there's a technology always wins, our knowledge base or our challenges. So actually, there's a technology evolved. Actually, I just started working in the cell biology immunology lab about 20 years ago, or 30 years ago, and then I published it like a T cell, NK cell analysis using this flow cytometer. I was kind of, lab manager of FACS core back then. And then actually it just leads to my master's thesis and actually doctoral dissertation. Although the doctoral dissertation was studying on one of the important immune markers called NF-kappa B that was aligned with my master's thesis. So NF-kappa B promoter polymorphism, some genetics on vascular function. So after postdoc training, I just joined Temple in 2009. And actually my very first publication at Temple in 2011 was about microparticle that is derived from endothelial cells, the vascular endothelial cells. And in collaboration with Dr. Fan, back then he was a director of our FACS core at Temple. And there's a support of my previous doctoral students. So it's published a couple of them. And then actually my attention goes to this concussion research, just our own Dr. Ryan Tierney. So Dr. Tierney and I kind of shared the same lab space in the basement of Pearson Hall. And then we just back and forth, a lot of chattings, and he's interested in genetics and I'm interested in some biomarkers in immunology. And actually there's like a new technology. You can really measure microparticles, the submicron particles using advanced flow cytometer. So actually I just decided to put my foot in this field. And then in collaboration with Dr. Tierney and his master's degree student that I co-advise, we just said, come up with some questions. So how to precisely assess the degree of head injury in a minimally invasive and cost-effective way, and then how to provide critical information to assist the clinical decision, whether or not you have to send athletes to the very expensive MRI, and how to develop an objective diagnostic and prognostic guideline, and how to better understand their biological processes. So we have used that already established in-lab repetitive subconcussive head injury model. Basically, you shoot the ball with JAG machines with identify the standardized speed and standardize the weight. And then actually the soccer players who have experienced more than 60 years were just standing about 20 yards away, and then they just receive the ball with their head for 10 times in 10 minutes. So every minute, balls are coming to them and they have to stay in the circle and they have to receive the ball with the head. Or for the control group, they have to receive the ball with chest or thigh. So we collect the sample, and using the flow cytometer, this is advanced model, basically from same company, Becton Dickinson. Actually, just analyze, we could analyze the extracellular vesicle containing proteins. And then we have two very interesting extracellular vesicle proteins that has shown to be changed following those 10 header sessions. So as you can see here, the timeline, there's a immediately after the 10 headers and 1 day recovery. And there are 2 markers out of tens of different markers that show some interesting data. So actually those 2, the biomarker, that is a positive biomarker, Claudin-3 and then MOG. Those are expressed in central nervous system cells, like endothelial cells and also oligodendrocytes. And those are the negative controls. So CD144 is called VE cadherin. That's universally expressed in any types of the vascular endothelial cell. And CD62E is expressed specifically in the endothelial cells with vascular inflammation. So we don't think that those 2 markers wouldn't change, but probably some of those markers originated from inside the brain. Somehow they can appear in the blood. Actually, as you well know, the blood circulation is pretty much isolated because of blood-brain barrier. So with those contact, with those, disruption of blood-brain barrier, those kind of cell dust from brain can come out to the circulating blood, and we will be able to detect those changes. So based on this data, actually we submit a grant to the NIH, and the first revision was that you have to expand your panel. So not just the 2 for like a multi-million-dollar grant. So actually we did a high throughput analysis that's called RNA sequencing by a previous doctoral student, Boa Kim. And then we identified the 23 most interested protein markers in CNS area. And also, after we got that grant, we just run with the same machine that we used to make the preliminary data. But in 2019, technology is evolving like lightning speed. So they invented a new type of flow cytometer called the spectral flow cytometer from Cytek Aurora. So actually this machine is also run by Dr. Fan, the same director, but now he moved to University of Maryland in Baltimore Medical School. What we can really separate is the really tiny small particles. So you can see those are size from 179 nanometers to about 1.3 microns. So you can reliably separate those small particles and count the number of proteins that is tagged in those nanoparticles. So actually we reanalyze our data with additional samples, and then we come up with beautiful data that was done by almost Dr. Megan Rath. And actually what we found was that, there was like GFAP containing EVs are changed, but it's not statistically significant. Although this is the pilot data with a small sample size. When we combine at least two different protein markers detection, then actually there is a more reliable curve. Actually, that does make sense to us that not a single biomarker can really predict what is going to be happening in their post MTBI in these sports athletes. So just we have turned our attention to different way of analysis of combination of different biomarkers. And also we use the other type of analysis is called a SIMOA or digital ELISA, which improve sensitivity about 1,000 times higher, greater than traditional ELISA. So it is a very, very precise machines in collaboration with Dr. Sylvia Sadi. We analyze some conventional markers that include like a GFAP, tau, UCLH1, and NFL. And actually, there are some more interesting biomarkers that we can analyze with this SIMO machine. So we have a lot of data. We just completed all the analysis. So this is the procedure of our work. First, we have to recruit the subject. And then we have to isolate EVs with the fresh blood, so every day. And then we have to run Aurora, that is the spectral flow cytometer in the University of Maryland in Baltimore. So actually my very committed, my doctoral students and also lab tech is traveling to Baltimore every twice a month to just analyzing this data. After we analyze those data, we get those data, and that's about halfway done. And then we have to analyze those dots on screen using software called the Flozo. And then it results in big data. So there are, originally it was a 23 biomarker. Now we have 27 new markers plus 4 conventional markers. So totaling 31 biomarkers. And there are around about 50 samples from our collaborator at the University of Delaware, about 50 control and experimental samples from Temple. So there are about 100 different subjects that we recruit over the course of 5 years, actually 3 years, because I have to exclude the COVID. And then there are 3 different time points. So totaling 300 data points. When you combine those 31 biomarkers, there are more than 1 billion cases. And it'll take about 34 years if you can analyze 1 case in a second, or you can use a 34 computer for 1 year. So there's no way we can just do it. So actually, we just collaborate with the Samsung AI department, and we had several very productive meetings so far. And now I'm waiting for NIH pre-approval. Then actually they have an idea that how their machine learning technology can help us to make a conclusion out of this big data. So that's our current status. There are so many different previous students who have been working in our this concussion projects. And I want to highlight those 3 people, my current members, Jacqueline and Megan, and our previous, the lab scientist, Suyong Che. That's it.
Great, Joon. Well, thank you very much for that overview. And what's really great to see with all of your slides is I think you all do a great job of highlighting not only the teams that are involved in this work, but the multidisciplinary nature of those teams. So I'm very appreciative of all of your slides at the end, highlighting all the work of not only our colleagues here, but our students and both past and current, who have been involved in that work. And, you know, it's also a great point of pride for me when I have my associate dean hat on, because, you know, I am really continually impressed by the amount of scholarship that we're producing that involves our students, and in some cases all the way down to advanced undergrads. And I think that that's a story that we don't always have an opportunity to highlight, but I'm glad that all of you did here. So if people have questions, please go ahead and throw them in the Q&A feature. We already have one, and I actually love this question because it dovetails with one that I wanted to ask as well. And the question says, when I got knocked out in a field hockey game, I couldn't see anything, but I could hear everyone around me. Could you describe what was going on in that moment? And Dr. Mansell, I'm going to start with you, because, you know, you would be the one who was kind of right there, and, you know, kind of potentially right at the field hockey player's side as she's still laying on the turf. So can you explain to all of us, and I think for those of us who are sports fans, we would love to know what's going on in that moment.
I saw Dr. McDevitt on mic so quickly that I think she wants to answer this one first.
Dr. McDevitt.
Being a field hockey player myself, and Dr. Wright, if you want to chime in. But so the hearing is stimulated differently than the ocular. So there could have been several different reasons why your vision was blurred. Going back to one of Dr. Wright's slides, you saw that during that acceleration, deceleration, you know, there were a lot of brain centers in the center of the brain that are disturbed, right? And going back to the pathophysiology, if we don't have that correct neuronal stimulation because of the hyperexcitotoxicity, difficult to get normal reflexes. So there are 3 different eye reflexes that allow you to focus and have proper eye vision and function. Many of which that we're seeing in the vestibulocular motor screening, such as the near point of convergence. If you aren't getting proper signaling, if that brain region that allows for that near point of convergence for you to be able to move your eyes and track your eyes to be able to see becomes problematic, you'll get nystagmus, abnormal saccadic vision, which could create then blurred vision. Other reflexes that could be dysfunctional during this time, especially acutely after that neuronal strain, could be the optokinetic reflex. So the gaze stabilization, you are unable to focus your gaze. You will get opsolopsia, where everything kind of looks like it's just dancing around, right? And you won't be able to focus your gaze or stable your gaze when you're trying to get up or move around. So I don't know, Geoff, did you have anything else? Or Dr. Wright, I'm sorry.
No, you covered a lot. I would say even just the hitting of, depending on how the brain gets hit. You remember that film that Dr. McDevitt showed where the brain was sloshed around, and the example in that video showed the brain, you've got your occipital lobe in the back of your head. If that crashes against your skull, you could have like a cortical blindness. So you'd completely not be able to see, possibly for a short period of time. Hopefully it just recovered after a couple of minutes, and then you'd have like these longer lasting symptoms that once your vision recovers, then you start having some of these reflex problems that Dr. McDevitt just referred to.
So a somewhat related question that I was curious about, and I think, again, for those of us who are sports fans, we probably had similar experience, but when we see that a football player, a hockey player, even a basketball player bangs their head, and the camera comes in close, and you can see that, their eyes have gone very wide and they look kind of disoriented in the moment, even before they start to take a step. Is that a clinical sign of a concussion, or is it really impossible to tell?
That's like the first sign, like the boxer referee who's saying, how many fingers am I holding? They're looking at him going like, if they can't answer that question, then that is a sign that they may be experiencing actually a loss of awareness without actually completely falling over. But I'm not a clinician, so I'll hand this over to some of the clinical people who probably have witnessed this firsthand.
I think that just goes to show you the value and importance of having an athletic trainer there on the sidelines, because that can be a first indication. And that's when we can pull all of the tools out of our clinical toolbox to make sure that we're doing the robust multimodal assessment to really take a look at what's going on there. And some of the NFL and some of the NCAA teams actually have like eyes in the sky, so concussion spotters, who are meant to look for some of those hits and some of those blows to identify, they'll call down to the sideline and say, you should really pull aside so-and-so and give them a full assessment.
Okay, great. So I'm going to turn to a question from Laura Vasallo, who's our director for the Office of Research Administration. And as she says, when I got knocked out while ice skating, I had a loss of memory, forgetting very huge events that happened in the previous days, things like Christmas Day and Christmas Eve dinner. About 15 minutes later, oh, I'm sorry. So she got knocked out. She had a loss of memory, but about 15 minutes later, all of those memories came flooding back at once. Do any of you have an explanation for what likely happened there?
Guessing without any extra clinical or any other data is, again, just like Dr. Wright said about, the brain sloshing, it sloshes forward and backwards. So you're getting the cognitive dysfunction from the frontal lobe being impacted and then stretched. So, that could be a reason why initially you get that initial kind of stun, right? That pathophysiological process happens. Your brain isn't synapsing correctly. And then, but once it starts to regain normal homeostasis and you can regain those normal synapsing, normal functioning, it could come back. But that's just a 100% guess.
Yeah, I think it does come down to, and I hate to say it this way, but the brain is so complex, but if you want to really break it down into smaller chunks, you can think of memory as the processing, the encoding, storage. And if you have stored it, then you need to retrieve it. So each one of those could represent a place where damage happens. So what's probably happening is that you had processed and then you stored and then you have it sitting there in storage, but somehow the process of retrieval has been affected. And so, Jane gave a nice talk about how these processes are affected with the loss of ATP and the influx of calcium. So if those processes are temporarily damaged, then the retrieval process might be damaged. And then once that process gets back and is working again, then those stored memories are just waiting there to be retrieved again as soon as your retrieval process is functioning again.
Actually, I can just add a little bit on the biological perspectives. So actually, the brain is one of the organs that is a lack of mitochondria except the cerebellum. So actually, there are not many mitochondria to produce ATP. And our body is designed to survive. So when you have injury, the energy should go to repair the injury, not for the memory. And then when you just get those injuries fade away, then you can use more energy to bring out or retrieve those memories. And you can just get some like fresh memory. Just my guts, just theory.
Like
Yeah.
So our last question actually comes from one of our students, which I think is a great one to end on because I think it's one we can all comment on a little bit. As someone who's in public health, but interested in epidemiology and head injury research and rehabilitation, what role could a public health major have in this work? She stumped you guys?
No, I just don't. My feeling is the epidemiologists are some of our best statisticians, and so I love statistics, but I also still go and talk to my epidemiologists and biostatisticians regularly because they just spend a lot of time really thinking about the stats, and so there are patterns, there are statistical questions, and Joon Park mentioned machine learning, which is a form of a statistical approach. A public health student, if you have a quantitative bent, then take lots of stats course. If you're interested in these epidemiological questions, I think that there are a lot of unanswered questions that just have to do with big data. There's so much data out there that we haven't even begun to tap into. And so each of us has probably more unpublished data than we care to admit. And we are just waiting for someone like a good epidemiologist or public health expert to come in and say, oh, I know what I can do with that.
I think that's an incredibly important point, for our students to hear. And we were having this discussion related to one of our studies just the other day that, you know, we have countless projects that could be led by students, and oftentimes it's the students in epi and biostatistics. You know, somebody working with our good colleague, Dr. Jingwei Wu, who I know is on the call, who, you know, is just a phenomenal resource to many of us. And then, you know, also our colleagues in health services health services administration and policy. You know, Dr. Mansell, as I mentioned early on, when you put up that graphic about the state laws, that's something that our colleagues there, our colleagues even in public health law policy in the law school could certainly help us with. But, you know, Dr. Gabriel Taju, among others in HSAP, is doing a lot with machine-based learning. And I can also tell you that from the university's perspective, they would love nothing more than to see collaborations across college. And so if it was something where one of our public health students took our data and then went and worked with Gabriel or our colleagues in computer science or engineering to do some of this machine-based learning and interpret the results from some of these smaller studies, it certainly would be smiled upon by high-level research administration within the university. So for the students who are listening, who were really inspired by what they heard, and I truly hope that you were, because I think this is such a phenomenal area of work in the college. I would really encourage you to reach out to the people who have been on this panel today. You know, send them an email saying I was really inspired or interested in what you do. I want to learn more about it. Most of us pride ourselves on being accessible that way, even though we know that that can be, you know, nerve-wracking for all of you to send those. So, you know, please do reach out. So let me take a moment to thank our panelists today. I think this has been a fantastic way for us to showcase some of the great work that we're doing in this area, and hopefully there's much more to come. And then a couple of housekeeping reminders. So next week's session, our colleagues from the Office of Community Engaged Research and Practice will actually be bringing in an outside speaker talking about the screening of colorectal cancer, which I think will be a great opportunity for our college community to see what's going on in the community about encouraging people to get screened for colon cancer. And again, we'll touch on issues of big data. And then 2 weeks later, on April 1st, we're actually having our last session of the semester with a little bit of a twist. We're going to do something having to do with spring cleaning. So we're having experts from Burrow and Birch who are going to present to us as we as faculty come back and learn, reacclimate ourselves to our labs about how we can clean up our labs and figure out that some things that have sat in drawers for 2 years, we could probably live without. And so I think there'll be a great thing for our faculty to think about as we come back to more normal operations in the coming weeks. And then 2 weeks from today, or 3 weeks from today, I'm sorry, I'm thrilled to say, will be our college's Research and Evidence-Based Practice Day. It's will be the final event of National Public Health Week. We again have a record turnout for our virtual poster presentation. My colleague Jackie Spitzer has done yet again a phenomenal job in coordinating this, getting the abstracts in, having them reviewed. We still need some judges, faculty, if you're willing to help us out. And I would be remiss if I also didn't thank Sam Yelman and Lorraine Ford and our colleagues in IT who once again are going to make this all be a seamless poster session for the third consecutive year. So with that, everyone enjoy the basketball this weekend, enjoy the nicer weather, and I look forward to seeing you next Friday. Take care, everyone.